Hello everyone, my name is Francisco Javier Chaparro. I am the Electrospinning Product Manager at Nanoscience Instruments. Nanoscience sells and supports a variety of scientific equipment in the North American region. Today I'll be presenting one of the multiple projects that we have been doing in collaboration with the Ohio State, and today's topic is Sintering-based porosity mapping of Electrospun PCL. The electrospinning technique is widely used thanks to its ability to produce nanoscale fibers, which allow for high level of porosity and surface area. These are crucial for applications like filtration. For example, filtration masks where nanofibers must be able to prevent virus-bearing particles with a size less than 100 nanometers. Lacking control over porosity could cause life-threatening losses if the filtration efficiency is lost. Another relevant example is in tissue engineering, where porosity is important to control cell infiltration, which have a direct success or failure of organ replacement. These types of products are generally characterized by the high level of porosity and large amounts of surface area. But the question of how much of the porosity across the product deposition is present still remains a question to be answered. Sample porosity can be measured in different ways. This includes image-based, where a small area of the surface of the spun sample is analyzed, typically by SEM. The disadvantage of this is that it is not representative of the sample as a whole. Taking multiple images, stacking them, and performing 3D modeling has been an alternative, but this is still not representative of that sample as a whole. Liquid intrusion is a second alternative where either mercury or non-mercury liquids are used to fill the sample, displacing the gas in all the pores, which are providing then a sense on the pore size and the porosity. We then have scanning laser micrometry, which is a robust technique capable of high precision, non-contact inline measurements commonly used in research labs and industry. Here, a laser beam is emitted from the transmitter, scans on a specific range at a constant speed. If an object is placed in this range, we can get dimensional information based on the time the laser was obstructed. This is an ideal way to measure delicate and soft tubular electrospun samples, as there is no risk for deformation. Meanwhile, knowing the apparent density and bulk density will only provide the overall porosity of the entire sample with no details on internal porosity variations. Towards this end, we generated an equation able to determine mass distribution within samples to compare it with dimensional profiles to then estimate the porosity distribution. By considering the diametric reading from a laser micrometry for the tubular As-spun and after sintering, we can obtain a diameter position profile across the rod length that can then be used to map the porosity of the sample. With a few assumptions, we have the thickness of the deposit, which is labeled as a capital H, radius of the underlying cylinder represented by a capital R, the span at an angle, and measured sample length represented by a lower L. With all this, we can then obtain an equation for the volume of the sample at a specific segment. Similarly, the volume of the densified sample can be obtained, and then in this case, the thickness is represented by a lowercase h. With these two, a simple porosity equation can then be developed which determines the porosity from the substrate radius, the as-deposit sample, and the densified thickness. In these images, you can see in the lower left how the Electrospun sample looks like, and then we can also see on the right side how the Electrospun sample looks after sintering, which is part of the key aspect of this development of the equation. In order to study this approach, we implemented the use of the Fluidnatec LE100 technology with tight control of temperature, relative humidity, and airflow by implementing the environmental control unit known as ECU. Globally, all Electrospun conditions were tightly controlled to maintain batch-to-batch consistency. Initially, we used a 5-way % PCL in HFIP solution, which is commonly used in tissue engineering applications these days. The right table shows all parameters used, which I'd like to point out that initial study was performed at inert conditions or at 0% relative humidity. Needle voltage was always maintained at positive 25 kilovolts, flow rate was used at 6 mil an hour, the needle collector distance was used at 20 centimeters, and the angle was at 45 degrees. The rotational speed of the mandrel, a 9.5mm mandrel, was 200 rpm, and the spun time was 15 minutes per sample. The main variable studied for this solution was the effect of the collector voltage on thickness and porosity, which we're going to be presenting in the next slides. Initial results show how a two-dimensional thickness versus position in the axial direction or across the length of the mandrel varies by applied collector voltage. At 0 kV, we see the typical As-spun sample morphology where the central position of the sample has a higher thickness up to 0.4 mm, which in this case is represented with a line in blue. When the sample is densified, significant shrinkage is observed to about 50 micrometer in the central location, which is represented with a red line. In the middle figure, we see the results for the positive 5 kilovolts applied in the collector. Not surprisingly, when this positive voltage is applied, repulsive forces are present, causing the thickness of the sample to be spread out more in the axial direction, and then after sintering, the distribution of the thickness has a similar trend. Now, if we look at the right figure, we see that upon closer inspection of porosity by using the developed equation, we can see that as we are expecting, the attracted negative voltage has a lower porosity throughout the sample, followed by the 0 kV or the grounded collector, and a repulsive positive 5 kV with a higher porosity with values that are going 90 to 91%. We also notice that the porosity tends to increase towards the edges of the deposition regardless of applied voltage, which is possibly due to better solvent removal across those areas. Now, in order to study the effect of shard density with porosity, the dye rose bengal was added in solution at an overall ratio of 50% PCL and 1% rose bengal. In order to maintain similar conditions, every single electrospun parameter was maintained, and the collector voltage that was studied was minus 5 kilovolts. The first change that we see with the addition of rose bengal is a more focused deposition in the central area of the mandrel with a thickness up to 0.5 mm as opposed to the previous sample without the rose bengal where the thickness was about 0.3 mm. Densification has a similar behavior, and the porosity is similar to the sample with only PCL. Meanwhile, one thing that we notice is a small dip in porosity in the central area, which we are attributing to possible solvent retention in that specific location. A key aspect of multi-needle spinning is how fibers from individual solutions will interact with each other and be collected throughout the sample area. To study this, we perform dual spinning using a PCL solution and a PCL rose bengal solution simultaneously, maintaining similar electrospinning conditions as before, using a minus 5 kV collector and then only varying the relative humidity. We also perform fluorescence analysis throughout the sample length to get a sense on the interaction of the two types of fibers and just to see if there is some really well uniformity in terms of like how the rose bengal containing solution distributed throughout the sample length. Dual electrospinning at 0% relative humidity shows a center line porosity of about 90% with visual uniformity sample deposition across the tube length and sample thickness. At 50% relative humidity, the central porosity drastically decreased all the way down to 0%, which is the image represented in the middle. As opposed to the central area, both humidities shows a higher porosity towards the edge of the sample. When looking at the thickness before and after sintering, this shows that there was relevantly no change in thickness in the central area, which contributed to the 0% porosity. A key aspect to note is that the sample has unimodal distribution at 0% relative humidity and bimodal shape at 50% relative humidity. Upon looking at the microstructure, we saw a large fiber structure present that likely result from collisions and agglomerations during spinning, allowing solvent retention in the area at 50% relative humidity. Although in the previous slide we saw an unimodal distribution of thickness at 0% relative humidity, when we analyzed the distribution of rose bengal through the fluorescence analysis, we found that the deposition of PCL and PCL rose bengal is not uniform. The blue areas represent PCL-rich containing areas and the orange represent PCL rose bengal rich areas across the length of the mandrel. At 0% relative humidity, we clearly see asymmetric distribution of rose bengal across the mandrel length. This suggests that at some point there is spatial separation between jets, establishing subsequent non-uniform pattern deposition. At 50% relative humidity, we saw pronounced compositional asymmetries with PCL-rich and PCL rose bengal rich peaks that correlates to the bimodal deposition we previously saw on the previous slide. These areas where PCL rose bengal are rich are expected to have higher residual solvent, causing the previously observed drop in porosity all the way down to 0%. This gives us a better idea that multi-needle deposition could be far from uniform, especially if a dual deposition of two different solutions with different charge densities are used. Up to this point we have shown a two-dimension profiling, but in order to have a better understanding of the porosity throughout the whole sample, a 3D profiling needs to be implemented. Here we show the profiling over the circumference of the rod, always measured individually before and after spinning and after sintering to obtain a 3D representation of the sample. The images in the right are for the mandrels represented in the angle of rotation from 0 to 360 degrees, its measured axial length through the sample deposition, and the obtained porosity by the applied voltage. In that image we see how the sample collected at 0 kV has a higher porosity than those of the -5 and -15 kV. A similar setup can be implemented in a flat Electrospun sample with a different sensor, something that is already available in the FluidNatech technology and especially for roll-to-roll deposition. In conclusion, the use of laser micrometry can be used to perform mapping on variations of electrospun porosity throughout the sample length. By globally controlling all electrospun parameters with the FluidNatech technology under tight environmental conditions and in combination with laser profiling, we can access to better process and explore different conditions that could be missing from current electrospun products. As expected, applied collector voltage has a clear effect on sample porosity. This knowledge can be used to target sample porosity based on application needs. Dual electrospinning causes large changes in porosity and microstructure according to the environmental condition used. This is why this is a key reason why employing an environmental control unit giving you the flexibility to increase and decrease temperature, relative humidity, and airflow is key for proper solvent removal and minimize solvent retention that can be affecting the sample microstructure and the porosity. Multi-needle deposition could have localized deposition depending on the solution process. Three-dimensional porosity map reconstructions allow us to quantitatively measure porosity across a wide area to better understand the sample properties and behavior needed based on application needs. Some of the future work that is to be explored are the effects of relative humidity on porosity using the FluidNatech technology. The beauty of electrospinning is that it allows us to have a variety of fiber diameter depending on the solvent identity. This is why using different solvents while maintaining the polymer identity is also another step to explore. One of the unique advantages of the FluidNatech technology is that we can implement a metrology system to measure thickness in real time, and even stop once the desired thickness is achieved. This will also be part of the future experimental analysis, especially as reproducibility is key when moving from an R&D project all the way to industrial production. Finally, a multi-needle system for large-scale production is another area that will be explored. With this, I'd like to thank you all for your time. If you have any questions that you'd like to reach out directly to me, here is my email. Feel free to reach out. Dr. Lanuri and Jisha will be able to help also during the Q&A section. Thank you. Bye.